Assessment of microbial safety in feed additives. EFSA evaluates microbial safety of two specific types of feed additives. Feed additives containing viable, non-genetically modified microorganisms, which act as the ingredient in the feed additive, and feed additives produced by fermentation using genetically modified or non-genetically modified microorganisms, in which the microorganism functions as the production strain. Certain requirements for specific scientific information need to be fulfilled. These requirements differ per type of microbial feed additive and per type of microorganism. A specific approach to assess the safety of microorganisms is the Qualified Presumption of Safety approach, or the QPS approach. A microorganism is suitable to be evaluated via the QPS approach if its taxonomic status is unequivocally established and the species is included in the QPS list. Additionally, the microorganism must comply with any qualifications set out in the most recent QPS list. Strains that fulfill these requirements are presumed safe for the target species, the consumer, and the environment, and no specific studies will need to be performed. The QPS concept can apply to genetically modified microbial production strains as well, under the conditions that the parental strain qualifies as QPS and the molecular characterization of the genetically modified microorganism does not give rise to any safety concerns. Whole genome sequencing plays an important role in the assessment of microbial safety. The information acquired with the whole genome sequence can cover several of the data requirements. Whole genome sequencing makes use of high-throughput sequencing technologies such as Illumina dye sequencing, pyrosequencing, and single-molecule real-time sequencing. To determine the complete DNA sequence of an organism's genome, including chromosomes and extrachromosomal genetic elements such as plasmids. Once the genome is sequenced and assembled, it needs to be annotated to identify genes in the genome and predict their function. EFSA requires the applicant to provide the whole genome sequence of their microorganism in the form of FASTA files. Besides the FASTA file, EFSA requires the following minimum set of information. Data obtained from whole genome sequencing is used to cover a series of requirements, including species and strain identification, characterization of genetic modifications, and search for sequences of concern. Whole genome sequencing data is mandatory for the identification of bacteria and yeasts, and is recommended for the identification of filamentous fungi. The microorganism must be identified to strain level. Once identified, the microbial strain should be deposited in an internationally recognized culture collection, preferably in the EU, and maintained by the culture collection for the authorized life of the additive. A certificate of deposition from the collection must be provided. This certificate specifies the accession number under which the strain is held. For fermentation products of genetically modified microorganisms, the genetic modification should be described, including the purpose of the genetic modification, the characteristics of the modified sequence, and the structure of the genetic modification. Preferably, this is done by comparing the whole genome sequence of the genetically modified microorganism with that of the non-modified parental strain. Whole genome sequencing data is also used for the identification of sequences of concern, in case the strain does not qualify for the QPS approach. Sequences contributing to antimicrobial susceptibility, toxigenicity, and pathogenicity need to be predicted based on the whole genome sequence data by identifying genes coding for known virulence factors or contributing to resistance to antimicrobials relevant to their use in humans and animals. This is done by comparing the whole genome sequence data to specific up-to-date databases. Besides genotypic identification of antimicrobial resistance, phenotypic tests should also be carried out for strains not qualifying for the QPS approach. Phenotypic testing must identify the minimum inhibitory concentration for a specific series of antimicrobials. When the MIC is below a certain cutoff value, then the microorganism is considered susceptible to the specific antimicrobial. Phenotypic tests should be performed to assess antimicrobial production. The inhibitory activity of culture supernatants needs to be tested against reference strains with a known susceptibility to a range of antimicrobials. The presence of DNA of the production strain should be tested in the final product if the production strain is genetically modified or if the production strain has acquired antimicrobial resistance genes. Antimicrobial resistance genes are considered acquired when a typically susceptible species is resistant to an antimicrobial. The presence of DNA is tested via PCR and must be done in at least 3 independent batches. Fermentation products must be tested for the absence of production strain. A well-described method for detection of viable cells of the production strain must be used. The method should be specific and at least 9 samples obtained from a minimum of 3 independent production batches. If your microbial feed additive complies with these mentioned requirements, your chances of receiving an EFSA clock stop will be reduced. Penentech specialises in feed additive authorisations, clock stops and renewals, so please contact us if you need any help with the application process.